In this video, we're going to talk about the post-lab discussion for chromatography, specifically paper chromatography, and some of the concepts involved in this lab. So let's talk about what the results showed. So by now, you should have already collected various different dyes and how these dyes traveled across the paper. To conceptualize this idea, I want you to think about what are some charges or forces that were involved in this specific experiment. We've already talked about dipole-dipole hydrogen bonds and London dispersion forces as being these intermolecular forces. And the whole point of this lab was to really understand how these forces interact with each other when the dyes begin to separate and as they're also traveling across the paper. So we have three different examples here to illustrate that concept. The first one being an ion exchange chromatography. Although we did not do this one in this specific lab, it kind of illustrates the idea of what happens when you have different charged beads and how they attract each other to what they call solvent flow. So if you imagine if I put in a positively charged beads inside this chromatography and I add in a solvent flow, and this could be water as an example, where it has a positive and a negative charge, there is going to be specifically certain types of ions that are going to get attracted to these beads. In this case, the negative charge to the positive charge. Now, it doesn't have to be water. It could be something that has both positive and a negative charge. For example, something like sodium. So if I add a positively charged bead in there, it's going to get attracted to that. That's what I call the ionic charge forces that are being attracted. Now the one that we will be using is going to be something similar to these three different ones. Paper chromatography is a little different because it has to do with affinity between the solvent and also between the paper. And in this specific case, you have specifically certain beads that proteins that attract themselves to these molecules. Now in this case, it could be paper, and the solvent flow in this specific case could be something that we're including with the mixtures. Now in this specific case, it's a little different because we're dealing with enzymes as an example. So enzymes have this ability to attach themselves to these, specifically these substrates, what they call them. So if you wanted to separate out substrates, and from the enzyme, you could technically do that or attach to them and be able to identify proteins. So chromatography is not just really great with mixtures, although we did do that today, but it also has different applications, specifically things like pharmaceuticals or protein purifications. We can also separate out ions in charges in specific water. There's another one called gel filtration chromatography, which again, same idea. All of these three follow that same concept, is that depending on charges, depending on shape, and also depending on how porous these beads could be, you can have that attachment. So I want you to really think about these examples while we're doing things like paper chromatography, where we're really looking for this ability for these ions to they're either charged or non-charged, to be able to attach themselves to the paper as it's going through what they call capillary action. This idea that liquid can travel up the paper because of this effect. Now, the concepts today that you were intended to really think about is this idea called capillary action. And capillary action is this whole piece of science that has to deal with porous materials, specifically the forces of adhesion, cohesion, and surface tension. Water has the ability to really interact with this and create capillary action because it actually allows molecules to really dissolve in it. Again, polar with polar, but also more importantly, it allows the ability for water to really stick to the paper and travel up using these three different forces. So I really want you to think about that whenever you think about water in terms of its ability to create surface tension. And surface tension has been one of the hallmarks of water. And again, by definition, surface tension is this ability for water to create a kind of almost like a net across at the top part, making some things like water drops or even things that have really, really tight hydrogen bonds. The other concept I want you to think about is this idea called solubility. And solubility, by definition, is the degree in which the solute dissolves in a solvent. And various different solvents have different degrees of solubility, as you guys tested today. Specifically things like the alcohol versus sodium chloride, and what concentration had higher solubility versus others that had low solubility. So when we're thinking about solubility, you always want to think about this idea called like dissolves like. In other words, we're looking at polarity versus nonpolar molecules, and also how do things change when you're increasing, for example, salt concentration versus increasing alcohol concentration. And there's going to be a different combinations that you could use. In certain cases, these mixtures could be separated using nonpolar solvents, and in other cases, it could be better using ionic bonds. Now, the separation of these solutions really depends on mobile phase and what they call the differential affinity. The ability for these solutes to be able to dissolve and be able to attach themselves to things like the paper. So here's a classic example of something where you can actually separate out the mixtures and see exactly the bands. So as you can see from here, this is the color of a Sharpie, right? So if I take a look at the color of a Sharpie, 4 dots, and I make 4 dots across, and I want to see what that color is going to look like, I can go ahead and identify the components of that mixture. So if I want to make black, brown, blue, whatever you want to call it, I can quickly figure out what dyes are inside. And if you take a look at examples, so we did not do other various different percentages, but we did run for sodium chloride, we did run several percentages regarding how much sodium chloride you have in there. So you should have seen a pattern. Did having higher concentrations of sodium chloride affect the movement? And the question is, why did it do that? In order to understand that question, you have to really dive into what's actually happening when you're dissolving salt in water. And in this specific case, we're dealing with isopropanol solutions, which is a nonpolar molecule, and it has a little bit of polarity in there as well. But more importantly, what's happening to the colors as it's running to the top. And so if you take a look at, as an example, with 50% isopropyl alcohol to a marker, what you want to really figure out is how far are the colors running to the top, and also more importantly is which colors are having a little trouble. Now in this specific case, all the markers went up at the same time, indicating that the mixtures were composed of the same type of molecules, except that they had a little bit of change to indicate the color. If you take a look at 100% alcohol, isopropyl alcohol, you can kind of see that now we're getting a little bit of a different pattern. So as you increase the concentration of the alcohol, you can kind of see that by 30 minutes, you could see these streaks begin to have a very different pattern. And that could be due to a couple of different things. First off, it could be the polarity of the molecules themselves. So if I'm looking at something like isopropanol, I have to take a look at the overall molecular structure of that molecule to tell me whether it's actually going to dissolve the liquid completely. Second thing I also have to know is what is the composition of the paper. So if I take a look at 100% at 30 minutes, you can see this purple streak go pretty far off. On the 55-minute, it's pretty close, but you can kind of see that the color is really fading on the 50%. So if I were to venture to guess exactly why that's happening, there could be various theories. But the most important theory has to be with the idea that as you're increasing the concentration of the solvent, there is going to be a different affinity due to the ability for that solvent to interact with the solute. And so as you're thinking about this, I want you to really hone in on the structural formulas for isopropyl alcohol and also of the mixtures. Now, just to kind of illustrate that point, if you take a look at what they call chromatograms, this is basically what happens when you have different concentrations of isopropanol alcohol with a Sharpie. Now, very similar to how you guys did your experiment, what you really want to take a look at is at 0% you can see that there is no run, 20% there's some run, and so on. And again, the ability for a color to run has a lot to do with how well it's dissolved in the mixture. And the second thing has to do with how much adhesion or cohesion you have with the paper. And so at a 100%, you can kind of see that's getting really close to the top. At 70%, we're about halfway, and at 50%, we're there. So there's something going on with that mixture that's allowing it to move higher. And again, it has to do with this idea of polarity and how well these molecules interact with each other. Now, moving along, you can kind of tell that, in specifically with different concentrations, you can indicate specifically what happens with solubility. And so what I do want to do is challenge you with specifically how well we can actually run this. Now, the first thing is paper is made from a cellulose particle. It's actually a carbohydrate. It's technically what plants make, for food and whatnot. And if you think about it, polarity on those molecules is generally speaking a nonpolar molecule. That's the reason why paper doesn't really dissolve in water. However, it does have nonpolar and polar regions. Now, if you change the solvent, and so if I take something like alcohol as an example, and I increase that concentration, the paper is going to have a different effect with the dyes themselves. Because one thing you also want to mention is that do these dyes, can they actually dissolve in water or in alcohol? And if you have a component that is polar versus something that's nonpolar, it's obviously going to dissolve much, much better in something that is polar. Like dissolves like. So if I take a look at this example one more time for paper chromatography, and let's take a look at Kool-Aid drinks as an example. And Kool-Aid drinks, for the most part, should be water soluble, which means that they are polar. If I take it in the lower concentrations, you can kind of see that the grape drink separated into the blue and red dyes. And in 100% isopropyl alcohol, it did not. So what that tells me is that Kool-Aid, not only is it soluble in water, but the fact is that if I were to choose what percentage I would use for perhaps maybe taking a look at that separation, what I would look for is something that has lower concentrations. Now, there's also another piece that you really want to take a look at specifically, and that has a lot to do with this idea of water, of why water is dissolving other substances. And again, there's this idea of hydrogen bonds, there is this idea of dipole-dipole movements that I want you to really explore and think about as you're doing it. So it's quite clear here that if I use 100% isopropyl alcohol, the separation is not as high. Something here with isopropanol in the midpoint, you can kind of see there's a little bit of separation, but obviously this is a poor choice of a solvent. Now, another idea here is different dyes have different solubilities. So if I take a look at this specific slide here about red dye as an example, this dye has a red and yellow separation. Yellow has low concentrations of isopropanol and less soluble in high concentrations. So obviously the colors definitely matter. If I take a look at the yellow color, it definitely behaves a little bit differently in low concentrations and less high concentrations. Red is slightly soluble in low concentrations and much more soluble in concentrations of higher than 100%. So that would indicate to me that the red dye has more nonpolar regions. And that would basically indicate the idea that it has dissolved in the solution and it has a little bit more adhesion to the paper itself as well. Okay. All right, guys. Well, hopefully this helps out with a little bit on the experiment. Make sure you review your data and analyze specifically what that factor is. Now, normally what they usually do is in these type of experiments is we study what they call the RF factor, or we call the retention factor. How far had the dye traveled relative to the solvent? So if the liquid is about halfway in, it's about a 50% or 0.50 retention factor. If it's all the way with the solvent, it is 100% or 1 retention factor, which means that it is completely dissolved in that solvent, and that allows it to be able to have that degree of separation from the other ones. All right.